And we're live. Okay, so now last framing question. How would eukaryotes have developed from prokaryotic cells? So first of all, we need to talk about what is true of eukaryotic cells. So eukaryotic cells have the following components. They have a nuclear envelope. That is, their genetic material is wrapped in an additional membrane. So in addition to the plasma membrane, bacteria, I know some of them having two of those membranes if they are gram-negative. But in addition to those membranes, the organelles, the structures inside of the cell, are also membrane-bound, especially the nucleus. And the nucleus is not just any membrane, but a very well-structured membrane to allow some materials to move out, some materials to move in, but most things to not be able to cross that membrane. You've got an incredibly important collection of material inside of that nucleus, being the genetic information, how you build the cell, and a way to control that. Mitochondria, and again, we said excavata for a long time. It was understood that they lacked mitochondria, and now it looks like they actually have just mitochondrial derivatives, that they've, for the most part, lost mitochondria because they don't need them. They're mostly parasitic forms, got an unlimited food supply, live in oxygen-poor environments. The mitochondria don't serve them well, so they don't form normal mitochondria. Instead, they form mitosomes that they use to do other tasks. Cytoskeleton, having a really complex protein scaffolding inside of the cell. These are much larger cells than prokaryotic cells, and to keep from basically collapsing, they have this really complex protein skeleton inside of the cell. Flagella and or cilia, and these aren't unique to eukaryotes per se. There are bacteria that have flagella, but the machinery is different. Okay, unique machinery for their flagella and their cilia. We talked about these are two of the ways in which our protist can respond and move in response to stimuli. And what do you call that? The movement in response to stimuli. Taxis. That's wonderful. So chromosomes. In prokaryotic cells, for the most part, the DNA tends to be circular and tends to be really small. In eukaryotic cells, the DNA is almost always in linear blocks called chromosomes that aren't just DNA, but also are wound around proteins. And so basically all of the genetic material in eukaryotic cells is stored a very specific way, is condensed a very specific way, forming chromosomes. You'll talk about that more in cell biology. Eukaryotic cells divide by mitosis. Prokaryotic cells do not. Again, you'll talk about that more in cell biology. And then sexual reproduction. That is unique to eukaryotic cells. There is some transfer of genetic material that you'll find in prokaryotic cells, but it is not like the sexual reproduction you find in eukaryotic cells. Okay, so here's what eukaryotic cells have, and so as you think of addressing this question, how would you get a eukaryotic cell from a prokaryotic one? You've got a list of features you need to develop, right? Anything that a eukaryote has that a prokaryote does not, you have to be able to develop it if you're going to move from a prokaryotic cell to a eukaryotic one. Make sense? Awesome. Okay, and so when you look at a eukaryotic cell, you see something that looks really interesting. It looks like two different cells put together. It looks like a larger cell and a smaller cell inside of it. There's no getting around that that's what it looks like. And so this idea or this concept is called endosymbiosis. One living cell inside of another living cell. So when you look at eukaryotic cells, I mean, this is what they look like. In fact, the structure that looks like a living cell within the cell looks a lot like an alpha proteobacterium. And so the interesting thing here is alpha proteobacteria, one of the taxonomic groups that were in that chart that I told you need to figure out a way to get comfortable with, which a week from today, you'll have an assessment that will see how comfortable you are with those taxonomic groups. Our first exam is a week from today. At some point today, I will post a study guide for the exam on Canvas. And so the structure of the exam is basically going to work this way. There'll be 4 blocks of matching questions. There'll be probably 5 multiple choice questions. And then there'll be 4 or 5 short answer questions. Okay, just to kind of put that out there. And so alpha proteobacteria, they use aerobic respiration, meaning they use oxygen to convert what they're metabolizing, those chemicals, into their energy molecules. And so the idea here is that the mitochondria, that should say mitochondrion, because then it's is subject-verb agreement. Man, that's terrible. This is plural. This is not. I apologize. You deserve better than that. So you can say the mitochondria are descendants, or the mitochondrion is a descendant of an alpha proteobacterium. And that's the idea here. And in many ways, a eukaryotic cell looks that way. It looks like that's the way it's actually built. And so some support to this idea. Mitochondria have a double membrane, which if you had one cell become incorporated into another bacterial cell, the way it would probably happen is some kind of endocytosis, okay, where the bacterium actually wraps around whatever that is, and so its membrane wraps around the original membrane, okay? And the mitochondria are double membraned. They've got 2 membranes surrounding them. The inner membrane has many complex folds. We call these cristae, and the really complex folds look like the membrane on an alpha proteobacterium. The structure of that inner membrane, which is the one that would have come from the bacterium, actually looks like the membranes of alpha proteobacterium. How much does it look like it? Depends on what you're looking for. You know, are you looking for similarities? You're looking for differences. Another conversation for another time. Mitochondria divide independently of the cell. They have their own DNA. They have their own ribosomes for making proteins from nucleic acids. They basically have their own stored information and their own machinery for turning that stored information into proteins. It's like, wow, it's really weird. Why does a structure within the cell have its own material and its own way to turn that material into proteins and actually divide independently of the cell? It really provides a lot of support to that idea that the mitochondrion is kind of a unique and separate living organism living within another unique and separate living organism. Okay? And so these are your supports or your arguments for the endosymbiotic theory, right? The first part of your homework was provide 3 arguments in favor of the endosymbiotic theory. Well, here are 3, okay, right here. Mitochondria double membrane, inner mitochondrial membrane looking like the membrane of an alpha proteobacterium. Mitochondria dividing independently of the cell. It's actually 5, containing their own DNA, containing their own ribosomes, right? So 5 arguments in favor. And then there's the question, and what the textbook makes an argument, is that plastids, like chloroplasts, developed basically a very similar way, okay, through this endosymbiosis. But instead of an alpha proteobacterium getting engulfed and becoming part of the other organism, you'd have some kind of photosynthetic bacterium, like a cyanobacterium. All right? So here are some diagrams, figures out of the text showing basically this process. Here you have your original eukaryote. And look at this. This is really interesting. And so we've got our proto-eukaryote. It doesn't have a nucleus. And then step 2, nucleus forms, right? And this is the defining mark of a eukaryotic cell, right? Is a membrane-bound nucleus versus a prokaryotic cell without the membrane-bound nucleus. Did you? And so finally, when you get to step 3, that's where you get the endosymbiosis, okay, forming the mitochondria. And so you have step 1 and step 2 not really getting into this endosymbiosis. And then when you get into step 3, you're getting the actual endosymbiosis, the formation of the mitochondria. Okay? And then from there, you get this now composite cell that looks in many ways like eukaryotic cells that we see now. Oh man, it's just, I mean, it lost power altogether. That's awesome. And then so with forming plastids, you're going to have a similar phenomenon where instead of engulfing an alpha proteobacterium, you're going to engulf some kind of photosynthetic cyanobacterium. Now, in formation of the chloroplast, you have basically two different options. You have primary, so primary endosymbiosis, cell engulfs cyanobacterium. And like engulfing the mitochondrion, you have two membranes around the chloroplast, which is true. Chloroplasts have a double membrane like mitochondria have a double membrane. Okay? And so this is primary. In secondary, a cell engulfs another eukaryote. Eukaryote with a chloroplast already. And so in this, you would expect to see some remnants of that original cell. And what's interesting is you find some photosynthetic organisms, some photosynthetic eukaryotes, that actually have what looks like a remnant of a nucleus inside of the chloroplast. Okay? And chloroplasts, like mitochondria, they have their own DNA. They replicate independently of the cell. They have their own machinery for turning genetic material into proteins. And so there's some good support in the way eukaryotic cells look, that you're looking at some kind of endosymbiosis to generate what our cells actually look like. All right? And so what I'd like for you to do right now, here's primary versus secondary. So primary, phagocytizing or engulfing a cyanobacterium. And then in secondary, phagocytizing or engulfing a eukaryotic organism. And then having a leftover, a remnant of the nucleus. And you find photosynthetic organisms that look like that. So there are your arguments in favor of this endosymbiotic theory. What I'd like for you to do now is I'd like for you to take a little bit of time just thinking about this. And I know you've put some things on your homework, and that's fine. You can draw from that whatever resources you use to make 3 arguments against endosymbiosis. But what I'd like for you to do is say, okay, assuming that that's where mitochondria and that's where chloroplasts actually did come from, right? So let's assume that that's actually true. What I'd like for you to say is, what do you still need in order to get a eukaryotic cell? If you have a prokaryotic cell that now has mitochondria, you still do not have a eukaryotic cell, right? You've got a prokaryotic cell with mitochondria. Noticing that in this, a step before even the endosymbiosis, you have to develop the nucleus, right? And so even if that's true for the origins of the mitochondria and the chloroplast, what do you still need to do in order to get a eukaryotic cell? All right, think about that. Take about a minute. Just think about that on your own. I'll let you know when that minute's up, and you can talk about it with those around you. Okay, 1 minute on your own, starting now. All right, go ahead and talk about it with those around you.
So That's able to read eukaryotic DNA, not prokaryotic.
And also when a cell develops, produces its own mitochondria as well, so that mitochondria is in the DNA.
Yeah, well, the mitochondria has its own DNA, but it also shares DNA with the nucleus of the cell. There's some processes that require both mitochondria.
That's fine. Thank you.
Mitochondria utilizes some of the
Don't be.
eukaryotic DNA to do stuff. Yeah. So it just needs.
All right. Okay. So we have a mitochondrion, right? And we have a chloroplast. So with those eukaryotic cells can get the maximum amount of energy production from whatever they're eating. Fantastic, right? They've got the mitochondrion, they can do that. With that, some eukaryotic cells that are photosynthetic, they've got a chloroplast, they can take sunlight and convert it into some kind of organic molecule and convert light energy into chemical energy. Fantastic. But you still have not explained how you get a eukaryotic cell, because what are you missing? Yeah, I mean, where on earth did the nucleus come from, right? I mean, that is the defining characteristic of what makes a eukaryotic cell a eukaryotic cell. And having a mitochondrion does not explain where the nucleus comes from. Yeah, what else?
It needs all its other membrane-bound organelles.
Yeah, so you need like endoplasmic reticula, you need Golgi bodies, right? You need lysosomes, you need peroxisomes, you need vacuoles in some cells. You need all kinds of membrane-bound organelles that this in no way gives you any explanation for how you get those structures, right? What else do we need? We need flagella with unique machinery that are not found in prokaryotic cells, right? It's kind of interesting, yeah.
The chromosomes, like the wrap.
Yeah, you need to change, basically not just develop the nucleus, but you have to entirely change the way that the genetic material is stored, right? Completely change the way the genetic material is stored. And what you'll see, if you have not already seen it, but not just the way it's stored is different, but the way you take it from storage to start to write, making copies, the machinery completely different. You've got to change machinery altogether. Okay? You need to grow in size, which that's not a huge challenge, right? It's fairly easy to get things to grow in size. If you take something that has a really short generation time, like a fruit fly, and you only allow the largest individuals to breed every generation, what you'll end up getting is fruit flies that are bigger on average than your fruit fly started as, which fruit flies are teeny, teeny, tiny, so bigger on average could still be a really teeny, tiny fly, but still you can produce changes in size without a lot of difficulty. I mean, look at a chihuahua, right? You want to change size, you're like, you know what, I want a dog that can fit in my pocket. Like, you can make it, right? Or I want a dog, you know what, that weighs 150 pounds, and when it jumps on people, like, it destroys their day, right? You can get that, right? Size is not really a huge issue, but then you need the scaffolding, that cytoskeleton that's going to support a much larger, more complex cell, and it doesn't just support it, keep it from collapsing, but it also provides roadways by which you can transport materials through a really large, really complex cell. So even assuming that endosymbiosis is right, and that's where the mitochondrion and this chloroplast come from, you have not in any way explained the origins of eukaryotic cells. Although it's like, you know, proclaimed as a triumph of explaining the eukaryotic origins. But you haven't explained eukaryotic origins at all, right? All you've explained is mitochondrial origins, yeah.
If it has to change how it works genetically, wouldn't that be considered changing its own kind?
Oh, well, I mean, who knows? I don't know. The machinery is really different. Okay, so now let's just get into arguments against endosymbiotic theory in general. So what I'd like for you to do is take a couple of minutes talking with those around you, share with them, because I know all of you submitted your homework assignment already, at least I assume so, because it said that there are 36 submissions, and there are 36 people in the class. So unless somebody submitted a whole bunch of times everybody in the class submitted this homework assignment. Okay? And so what I want you to do is share with those around you your arguments against endosymbiosis. And here we're going to focus and we're going to talk about arguments specifically against this being the origins of mitochondria and these being against the origins of chloroplasts. All right, take a couple of minutes starting now.
So, I wrote is the amount of DNA that would have had to be transferred between the cell and the mitochondria for it to bond with the cell would have to have been fixed to allow both of them to work together and then be perfect.
Exactly. The other one is naturally supposed to, if a cell involves another organism, it's to fulfill it.
Yeah.
So how in the world was that suspended for?
Yeah. I also put another fact is that both cells seem like they can only work when they are paired with each other versus both for them to be apart.
Seems rather.
Yeah. If let's say the cell that the mitochondria went into had different organelles already in it, those that couldn't even function without the mitochondria. So we need not to help drive the function.
So it's like, what came first, the mitochondria or the Golgi bodies?
Yeah.
They need each other. They can't, one cannot go without the other one. Is that basically what you're saying?
Kind of, yeah.
Okay.
Yeah.
That's a problem. So there are three of them out there.
Yeah. And like I said too, like you said, because when you split, the mitochondria split too. They go through their own.
All right. What do we got? Let's talk about some arguments against this even being the origins of mitochondria.
One of the things I found was that mitochondria membranes aren't made of peptidoglycan like the bacteria are.
Certainly not. Yeah, and so the inner membrane of the mitochondrion looks like the membrane of an alpha proteobacterium, but there are no proteoglycans, which is kind of like a telltale chemistry. And I know that's in the cell wall and not actually in the cell membrane, but you have none of the telltale chemistry of any remnants of that kind of a cell wall. Now, if your original eukaryote developed 2 billion years ago, is it possible that over 2 billion years you can lose that? Absolutely. Yeah. What else? Yeah.
I just noticed, it's all based on the assumption that prokaryotes were the earliest life forms.
Oh, sure. So if that's not true...
Yeah, we talked about, when stuff gets fossilized, it looks simpler. It gets fossilized simpler anyway. So we
Right.
don't know if
Yeah, and a lot of our fossils of prokaryotes are like fossilized remains of the biofilms, right?
Yeah.
And so it's like you don't really have a ton of fossilized cells. You have fossilized remains of like these complex biofilms that bacteria produced, right? So that's a challenge. That's an issue. Yeah, and so it's based on the assumption that prokaryotic cells predate eukaryotic cells. That's a bit of an issue. Yeah.
Jack and I came with three.
Okay.
First of all, you would have to.
Ambitious.
I like being ambitious. You have to completely rewrite the DNA of the nucleus of the host cell because they work together with the DNA of the mitochondria.
They certainly do. So mitochondria divide independently and they have their own DNA, but it is not all of the DNA necessary to make the mitochondria function, nor all of the DNA necessary to help the mitochondria divide.
Right.
Yeah.
Typically, when a cell consumes another organism or another cell, it is to actually consume it for energy and to use it. It usually breaks it down.
Right.
So for some odd reason, there's a fluke in the system, and it did not actually digest this other cell.
Right. You needed some kind of foresight there, right?
Right.
Where it's like, man, it'd be really nice if instead of eating this organism that I just started the process of eating, I let it live, right? It's like, you know what? Wouldn't it be fantastic if I just, kind of swallowed a plant seed and I knew if I left it there, like I could potentially produce a plant that, over time it can start to function independently of light since there's no light inside of my gut, and maybe it can grow me fruit, right? And I can become kind of like a self-sustaining, right? It requires some kind of a foresight into, man, it's going to be really nice. And so one of the assumptions of science we talked about our very first time together, and I know this was a long time ago, and you've probably forgotten that interaction. I haven't, because I remember all of my interactions with you. Now I'm sorry. Oh, yeah. So one of the assumptions of science is that the way things operate is an indication of the way they have always operated and the way that they always will operate. This idea of uniformitarianism. So it's interesting. If if this happens, right, if organisms can do this, can engulf something, can start the process of eating it and then not finish eating it, we ought to see organisms still doing that. But living bacteria don't behave this way. They don't do this. So it's interesting. We need some maybe unknown mechanism that's no longer at play, or at least under our conditions. So, yeah, what was your third?
And the third one is, what came first? The mitochondria or the Golgi bodies and the other organelles, because they rely on each other. You need the mitochondria for extra energy, etc., to support.
Right, you can get away from that issue if the mitochondria at one point had all of the DNA necessary for its function, right? Which now it does not. Some of the materials that function in the mitochondrion are in the nucleus, right? Some of the genes for that is in that. Some of that uses the cell's machinery, which means you need some way to package those proteins and ship them to the mitochondrion, which, yes, you would need the Golgi apparatus. But if when the mitochondrion came, they had all their genetic material, then you could get around that issue. But then you have another issue of where's the machinery to actually move the genetic material from the mitochondrion into the nucleus. And it's not just a matter of moving genetic material. I'm going to tell you this, you should write this down. You should absolutely write this down. You should circle it, do whatever else you do in order to make sure you know that this is really, really important. Okay? You ready? So the genetic code, how you actually translate from nucleic acid to protein, is different in the mitochondrion than it is in the nucleus. Okay? So the way you translate from nucleic acid into protein, which that's called the genetic code, it's different in the mitochondrion than it is in the nucleus. Which means you don't just need a mechanism to actually move DNA from the mitochondria into the nucleus, but you need a mechanism to change the way it's translated. So you're going to adopt the information and read it in a completely different way. I mean, that's fantastic. That's not even, even we don't do things that way, right? If you want to take something and adopt some information, you better find somebody that knows how to read that, right? And knows how to interpret that. You can't take some piece of literature written in a different language and translate it the same you would translating from Spanish into English, right? It just doesn't make sense. It just doesn't make sense. Yeah.
So by genetic code, you mean the way that the DNA unravels and then
Well, so it's getting beyond that. I mean, that machinery is way different too. But just in terms of so if you're going to translate, right? If you're going to translate, how many of you speak more than one language? Okay. And so if you're going to translate, right, you're not just going to think in the separate languages, but you're actually going to translate. You have to go in and you have to find basic, trends or basic patterns by which you can then go and translate. For instance, if you're going to take a verb from Spanish and you're going to translate it into English, right, you have to kind of figure out what the actual verb is, not necessarily the conjugated form of it, right, but what the actual verb is so that you can figure out the corresponding verb in English and then worry about the conjugation from there. Okay? And so in the genetic code, what you're doing is you're taking a language that's inside of the nucleic acids, which basically is a string of bases, A's, G's, T's, and C's, and then translating that into a different language that proteins are written in, which is a string of amino acids like alanine, lysine, isoleucine, leucine, glutamate, glycine, right? Yeah. So you have to completely change from one word to a corresponding word in the protein language. And so the way you actually do that translation is different for nuclear DNA than it is for mitochondrial DNA, which is why mitochondria have their own ribosomes. Because if they're going to make their proteins, they translate in a different way. Their genetic code is different. So nuclear ribosomes would not work inside the mitochondria.
It's like its own culture inside of a cell.
Oh, it's way more than its own culture. It's its own entity. Yeah. Which is why it's it looks like it's a separate organism altogether. And
that's all the more reason to believe that it could be incompatible.
Well, it's kind of, so something that would be sort of an analogy, and I know all analogies break down at a certain point, but it's kind of, there's some similarities to the gut bacteria we have, where it's like, yeah, they're their own organism and they're genetically distinct, but we're not human without them, right? And so if you want to make the argument that the mitochondrion is its own entity apart from the cell, that's fine, but it still is together a single functioning unit, right? And you've got a lot of questions. And when Lynn Margulis originally popularized this, the textbook gave like one sentence saying it wasn't originally accepted, but now it's kind of the explanation for how eukaryotic cells came to be, which is absurd because it doesn't explain origins of eukaryotic cells at all. It just explains the origin of mitochondria. But I mean, it was a decade before anybody even thought anything of it. And the reason why is because there's just a lot of obstacles that you have to overcome. But here's the issue that you came to. What better explanation do we have? Well, we don't have one, unless, of course, they were designed, and we don't like that explanation. And so then this is kind of like the best explanation. Does it look like eukaryotic cells have separate entities that live inside them with their own DNA, their own replication? Yeah, it does. Does it look like maybe that could have at one point been its own separate living organism? Yeah, it does, but you've got some huge obstacles you have to overcome to make that work. Yeah. Any other ideas? Arguments against? I mean, the biggest one is just the idea that you are in no way showing the origins of eukaryotic cells. Like, even if you can make a good cell that this is where mitochondria came from, we still have no idea where eukaryotic cells came from. No explanation for how you get a nucleus, no explanation for how you get the cytoskeleton, the unique machinery, no explanation for how you get the other organelles and their unique machinery. So you just have done really nothing, right? It's like, well, where did this house come from, right? You walk into a house, you're like, wow, this is fantastic. This house is so awesome. Say it's a smart home, right? You can pull out your phone, turn on all the lights, turn off lights, control your thermostat, whatever. Get into your crock pot, turn that sucker on so it starts to make your pot roast, right? I mean, this house is just fantastic. And you're like, man, how did we get this from like this, yurt? And if you like yurts, I'm sorry, but like you've got this yurt, which is basically just a big old dome tent that's very simple, like it doesn't even have windows, it's just an opening in the canvas, right? And you're like, okay, well, how do we get this from this? And you're like, well, inside of this smart house, we actually have this structure that kind of looks like a yurt, and it kind of functions like a yurt, so this is where the smart house came from, right? You're like, well, no, that may be like where that structure inside of the house came from, and I don't know why you'd have something that looks like a yurt inside of your smart home. My story broke down, I know, but anyways, but that's kind of what you're doing with this illustration. You're like, oh man, like this, really smart refrigerator inside here kind of looks, functions like an icebox, like a cooler. This is a better one. And you're like, okay, cooler keeps food cold, this keeps food cold. Yeah, this may have been where the refrigerator came from. Well, we've got our whole smart house, right? Just, given enough time, your cooler's going to become a refrigerator, and that refrigerator is going to give this house the ability, the energy requirements necessary to develop all those other wonderful things. And it's just, man, it's not that simple. It's not that simple. You haven't explained the origins of eukaryotic cells, and you very unlikely have even explained the origins of mitochondria. Just too many mechanistic issues. Too many machinery that just aren't there. Too many questions, yeah.
So if we were to say all this to an evolutionary cellular biologist, how would they respond?
Well, I think most of them are honest that the endosymbiotic theory has some major holes and some major flaws. And so, they would probably, I think most of them would respond in a snarky way and come up with some question that you couldn't answer and say, yeah, you're an idiot, right? You have no business to even ask those kinds of questions. Right?
They wouldn't have an intelligent way to respond.
So I'll give you an example. Hey, class is not done yet, right? This class goes till 12:40. I hear you all packing up. Don't do that. It's not nice. It's not nice. It's Friday. Don't ruin my Friday by trying to get away from me faster than you ought to. Okay? This is our time together. Okay, so there's a group of geologists that go to the national meetings every year. It's called the Geological Society of America, and it's just huge. I mean, thousands of geologists all in the same place, a whole bunch of aspiring Indiana Jones, like coming in, giving their talks, and it's fantastic. There's a whole group of people there that are young Earth creationists, right? And they go there and they have their own like meeting. They present in secular. Anyways, like it's just an entity within an entity.
Like the mitochondria.
Yeah. And so one of them I know really well, and he went to a talk from Stephen Jay Gould, who's now deceased, but one of the most well-known evolutionary biologists and geologists of the 20th century. And so he was presenting on basically the fossil record, and you've got a really big issue in the fossil record, and that is what's called the Cambrian Explosion, where you just end up with basically almost all living groups of animals all show up at the same exact time with no intermediates, no explanation of where they came from. They just all show up at the same exact time, which having a global flood is a great explanation for why that happens, right? And so he asks them, he says, Dr. Gould, he says, you've got this Cambrian explosion. Before he could even finish his question, he said, don't bring up the Cambrian explosion. What are you, some kind of like young Earth creationist? And just ignored him and went on to another question. And here's the issue. It's like, there are big questions that need to be answered. And the same thing for us. Like, there are big questions, or at least for me, anyways, there are big questions that need to be answered and are tough to answer, right? And are hard to figure out. But the thing is like we need to think about those and not just kind of keep it simple. But the problem is if you make your model really, really simple, by the time you've got buy-in, right? Or by the time it starts to get complex and you see how hard everything would actually be to make that happen, you've already bought into the paradigm, right? So it doesn't, you'll reconcile whatever doesn't fit with your model because it's already your model, it's already your paradigm. And so the thing is like you got to get somebody to adopt the paradigm before they realize how complex it is. So, all right, now our time is over. Have a wonderful weekend. Be safe. Make good decisions.
And that's interesting because scientists, they have it, at least anyways, I thought, that you have it grounded